on to the main event. I am delighted to introduce John Meiklejohn. He is a member of the board of directors of the Massachusetts and Rhode Island chapter of the American Chestnut Foundation. Since 2005, he has been the volunteer manager of a third-generation backcross breeding orchard, and more recently, a fourth-generation orchard on his property in Granville, Mass. And I'm going to turn it over to John. If you have questions, John's willing to answer them during the presentation, so I will be monitoring the chat, and also you can raise your hand, and there should be, if you look at the bottom of your screen, there's a reactions button, and if you click on that, there is a raise hand reaction. So if you can't figure that out, just put it in the chat and I will flag it so John knows to answer the question. Okay, John, you're on.
Thank you, Renee. Can you hear me okay? Coming through all right? Okay, great. Well, thanks both to Renee and also to the Pollinator Network of NOFA's Mass chapter for inviting me to have an opportunity to share with you the story of the American chestnut and the efforts of the American Chestnut Foundation to restore it. This is a story and an unfolding scientific adventure that I always enjoy being able to share, and I have been part of the Mass Rhode Island chapter's efforts since about 2005. And as Renee said, have a couple of orchards that I manage on my property in Granville. Before we get into the PowerPoint presentation, just want to ask any of you, and if you are able to just maybe raise your hand, so to speak, by coming on the video. If any of you, I'm curious if any of you have visited one of the orchards in either Massachusetts or Rhode Island. There are a lot of research orchards, more than 30. I just wonder if anybody's visited one, had a chance to have a tour or work on a work party. I see Ellen is raising her hand. Lloyd, Isabel, oh good, some of you. Could one of you just tell me what orchard you visited? Who was it? If you remember who was managing it.
So this is Ellen. Bruce Spencer in New Salem.
Sure.
And also I've had some nuts brought to me by Mike Meiklejohn, who I think you probably know if you're a member of the Chestnut Foundation.
I do indeed.
And I have raised them and planted them in my own orchard, and some of them are still alive. I'm not quite sure what to do with them right now, but I probably have 8 of those that are about 5 years old now, maybe 6. Yeah.
And I would imagine those were pure wild Americans. Is that correct?
You know, Mike wasn't sure what they were. He got them at the American Chestnut Foundation meeting about 6 years ago. So, yeah. And Bruce gave me a couple of trees several years ago that I think were actually some degree of backbred, but both of them have got some blight. They're not in my orchard, they're in my other woods.
Yeah. Okay, very good. Well, if I had time, I would ask all of you, where have you been? What your experience has been just having a chance to visit an orchard. But it is possible to visit the orchards. The orchard in Granville that I oversee, I've given a number of tours. We also have work parties at not only my orchard, but other orchards. So if after you hear this talk tonight, if any of you are interested in kind of learning a little bit more and seeing the trees on the ground and in their research orchards and learn more about it, just reach out either to me or reach out to Kathy Desjardins, who's here tonight. She's our chapter president, and the information for contacting us is at the end of the PowerPoint, and also obviously you could find out through Renee. So I do want to take questions as we go along. There'll still be time at the end for questions, but it's much better to take them as we go. It's just a better flow. And Renee's going to monitor the chat for folks raising their hands. One last thing before we get going, I have a story to share I've never shared before on one of these chestnut chats, and it's about the chestnut bee. And I thought, my gosh, if I'm talking to a pollinator network group I can't resist sharing the story of the chestnut bee, which was thought to be extinct. So it's not built into my talk, but I have a kind of summary of a news story about the chestnut bee. And if I don't mention it, just bring it up to make sure that I remember to share it with you. So let me go to the screen share here. And I'll pull this up. Let's see. Now, can you all see that okay, Renee? Is that showing full screen? Okay, good. So this is I don't know how many of you know the history of the American chestnut. I'm going to take you through just the sort of an overview of its history, in our culture and in the ecosystem, and then we'll get into the scientific ongoing restoration efforts. But this was what you might call a mighty giant. Here you can see some pictures of typical chestnut trees in the eastern forest of the United States, back in the day. And the story is, on the one hand, a tragic one. This was a tragic loss of a beloved tree within 2 generations in the first half of the 20th century. But it's also a story of hope, because there are these ongoing efforts to bring it back. And it's a story of hope in the sense that everyday citizens like yourselves and myself, who don't necessarily have a scientific background, but have an interest in the ecology and healthy ecosystems, there's an opportunity here to play a role in bringing back this mighty giant. And that indeed is the exact mission of the American Chestnut Foundation from its beginning 40 years ago. Its mission is to return the American chestnut, a blight-resistant version of it, to its native range. And as you'll see in a map in a moment, that native range ran from Mississippi all the way through the Appalachians up to southern Maine. Let's go to the next one. And here, in fact, you can see that map. Here's the native range in dark green, and the heart of it running right through the Appalachian Mountains, but also very much up into our territory in southern New England, and up into parts of New Hampshire and Vermont and southern Maine. They do the 1 in 4 trees. I would say that might be 1 in 4 hardwood trees as opposed to 1 in 4 trees. I'm a little dubious of that particular stat. But this was a dominant tree. It became a dominant tree in part because of its growth characteristics, and we'll touch on those here in this screen.
John, we do have a question about how the range and niche overlap with oaks.
I don't actually have an answer to that. I know in Maine, for example, we have a place in northern Maine, and we don't find any oaks there. But in southern Maine, I suspect you probably do. Someone else on the call actually may know more about that than I do. But certainly through the heart of this range, there are a whole variety of oaks that some more common to our area, like the red oak and the white oak. We have those on our property. And as you head south, I'm less familiar with the oak species that are down there, subspecies. The others may know more. If they want to put a note in the chat for people, they're certainly welcome to do that. But even if it's just 1 in 4, the hardwood trees, in the heart of its range, that's an indication of one of the ways in which it was an iconic and dominant tree. And it could grow to immense height. It certainly was a canopy tree.
Did you see my message?
Did somebody put a message in the chat, Renee? Because I can't see the chat.
No.
Okay.
Oh,
Did
I'm
somebody
sorry.
want to speak about that message?
Ellen, did you have a question?
I'm sorry, I was yelling at my daughter. I'm going to mute myself, sorry.
Okay. Yeah, if everybody isn't muted, just go ahead and mute yourself. And when you want to raise your digital hand, Renee or I will see that, or you can put a comment in the chat, and that way we know that you have something to share or a question to raise. These trees were very long-lived. They could live up to 400 or 500 years, and remarkably, they could be 14 to 15 feet in diameter. The biggest one that I know of that was ever identified was in North or South Carolina. It was something like 14 feet in diameter. I mean, it's just inconceivable in this part of the country. But you can see, given that kind of dimension, you can understand why it was nicknamed the redwood of the East. You remember out west or seen pictures of redwoods, well, they get to be that big. So the chestnut was a tree that provided a wide range of gifts, so to speak, both to the human community and to the wildlife of the eastern forest. It was a steady, reliable food source for so many animals in the eastern forests, not just deer and turkeys that you see here, but bear, the variety of birds, partridge, the blue jays, squirrels, and the carrying capacity of the eastern forest. When the chestnuts were thriving and before the blight came along, the carrying capacity of the forest for wildlife was greater. And that's because chestnuts produced a mast crop every year. That's to say a nut crop, whereas other trees that produced a mast crop, hickories, oaks, they year to year, their mast crop would vary. Some years it would be substantial, other years limited, and some years no crop to speak of at all. So when the blight came along and wiped out this steady basic source of food, the carrying capacity of wildlife in the forest declined. And there are charts, I don't have a chart to show you, but there are charts that they track that. And then for the human community, there are an enormous range of uses. The chestnuts were put to obviously for building and construction, but chestnut is very high in tannin, and so it was useful in the leather industry. And the farms and the households, homesteads, all throughout the Appalachian region in particular, they would rely on that crop. They both for their domestic animals, like their pigs, but also for themselves. Obviously, it was a food source for them. Plus, it was also a cash crop for them. They would collect the chestnuts, take them into the nearby city, and sell them to street vendors to get cash money. They called it shoe money. Many folks in Appalachia did shoe money in order to buy the shoes and clothes for their children and for themselves. So it was a tree that had so many versatile ways of contributing and providing gifts to the wildlife community and to us. So let's go to here, and then the blight hit, and it was about 1904 when it was the Bronx Zoo's forester. His name was Herman Merkel. He was the first one to identify this disease that had never been seen before. He didn't know what it was, but it very quickly in the next few years began to spread at a rate of a 50-mile radius, the north, south, and westward. And you can see on this map how rapidly it spread. By 1914, it was out into Pennsylvania. and down into Maryland. By the 1930s, it had covered all the way down to areas of West Virginia and Virginia. And by 1950, actually all the way down to the very southern end of the range, native range of the chestnut. And it was, very quickly by 1914, it had reached into New England. And the result was the collapse of a keystone species. As many of you know, a keystone species is one upon which other species in a particular ecosystem depend. And it led to a domino effect, both on the ecology and the economy of the area, especially where it was a prime crop and the commodity communities in the heartland, the mountain families in Appalachia, they were left with less game to eat, less food for their domestic animals, the elimination of that cash crop I talked about and their shoe money. It was a devastating loss, and it happened all within basically 2 generations. And we're distant enough from it now, there's hardly anyone, who remembers it and can give us a personal sense of loss that they felt. But when you read stories of people who lived through that time, and particularly folks lived in the Appalachian Mountains, they're painful to read. They lost somewhere around 4 billion trees as a result of this blight. And 4 billion trees, it's just hard to wrap our brain around that. But if you think of the state of New York, and if you think of New York State being completely forested, that would be about 4 billion trees. And this picture on the left, in some of the heart of the area of the chestnuts region, native region, these trees, it wasn't just 1 in 4. You could have acres that were basically, they predominantly or entirely chestnut. And so you can see here a picture of, these are all chestnuts that are standing dead ghosts. And imagine living in Massachusetts, as if we were, maybe in Tennessee, and our forest around our home looked like that, and what it would feel like to lose such a dominant presence on the landscape. It's just heartrending. So what did it? Well, here's a culprit. The blight is a fungus. The chestnut blight is a fungus. The official name, taxonomic name, is Cryptphonectria parasitica. And the picture on the left is an example of the cankers that develop on a chestnut tree. The characteristic, the reddish, the spores, and you can see the cankers spreading up and down the 2 trunks of this particular tree. The blight, the fungus, the way it functions, it's airborne, and it will enter the tree through cracks that are just the normal, the little fissures on the surface of the bark. But also, if there are wounds on the bark, they can enter the tree through the bark, through the wounds. And they do form a canker quite quickly that, if unimpeded, will eventually girdle the tree. It attacks the cambium layer and will kill the cambium layer of the tree, so that the tree cannot carry any water, or is restricted from carrying any water or nutrients to any part of the tree above where the canker is. So the tree above that canker site will die. That is an interesting chart of chestnut survival. The American chestnut is functionally extinct, but it's not officially listed as threatened or endangered or extinct. And there are still the trees surviving throughout the range. And you can see that here. This was the data developed back in 2010. And the most, not surprisingly, the most stems per square and the kilometer are right in the heart of the native range of the chestnut. Also notice the dark brown up here in central Massachusetts. So there are probably several 100 million chestnut stems around. You can find them. My guess is if we had time, asked you to raise your hands. Some of you have certainly found chestnuts in the forest here in Massachusetts and southern New England. But in general, they aren't much larger than about 1 inch DBH, 1 inch the diameter at chest height. And most of them don't flower or produce nuts, but nevertheless, and this is an important piece in the restoration of the chestnut, we do find, and you can find chestnuts that are reproductively mature, and you can collect nuts from them. And in fact, that is, there are a couple of folks in the Mass Rhode Island chapter where they devote themselves every season to collecting wild American nuts, the wild American chestnuts. And we send them down to Meadowview for the research purposes. We propagate them, and I will tell you later some of what we're doing in a very systematic way with the nuts that we are able to collect. So how do you identify them? Well, the best time of year is June and July, when the trees come into flower. And by the way, that's a fairly late period for flowering in the spring for most trees. Most trees flower earlier than that. And as a result, that may be one reason, this is somewhat of a guess on my part, but that late blooming pattern may be one reason why chestnuts did become dominant, because they wouldn't be so vulnerable to late frost. And late frost could set back trees, of course, as well as other plants. So that late blooming habit might have benefited them over time. On the left, they are the male catkins. And as you can see, they look a little bit like pipe cleaners. And they have a very distinct, the odor that's a little like bleach. And the female flowers, on the right, you can see a mature female flower that's turned into a burr. The female flowers, when they have ripened and gotten to this stage that you're looking at, they look a little bit like giant green sea urchins, and they are super prickly. If you found one that was opening and you could see the nuts and you wanted to get to that nut and you didn't have gloves on, you would try to pick the burr only once with your bare hand. You would not do it again. And I've learned I wear winter gloves, lined winter gloves, when I go harvesting in the fall, and that would recommend anybody else do the same. So in terms of identifying them, here you can see both sampling of the nuts of the 4 different main varieties of chestnuts, and also the leaf patterns. The leaf on the left is an American. You can see it's kind of canoe shaped and has very distinct scalloped edge that kind of looks like a wave, a crashing wave. But the scalloping along the edges is more distinct than any of the leaves that you see here. And the one that's just to the right of it is a Chinese chestnut leaf. It also has scalloping, but you notice the scalloping is much more abated and not as distinct. And the shape is different. It's more the oblong and not so canoe shaped. The nuts, by the way, you really can't identify a tree by the nut. But the smallest nut, the one on the left, the smallest nuts tend to be the Americans, and the smallest nuts are the sweetest nuts. So the Americans are the sweetest of all four varieties. The ones that we find in the Thanksgiving time, Christmas time is when we find chestnuts in the market. And when I find them, I always look to see, well, are these Chinese? Are these European? They hardly ever tell you. But most of the time, I think they're European chestnuts. That would be my guess. So here's our mission to return the chestnut to its native range. But the tree itself doesn't have any resistance to the blight. And the Chinese chestnut, does, because the Chinese chestnut evolved in Asia with this blight that is native to Asia. The fungus is native to Asia. So one would reasonably ask, well, why not they take the Chinese chestnut that already has blight resistance, instead of going through this long process of trying to breed in blight resistance through back-cross breeding and other techniques? Why not just use the Chinese chestnut? And here's the reason. If you look on the right, you see an example picture of a tree that healthy tree. But it's kind of sort of apple tree shaped. It's chubby, and it is resistant to the blight. Grows to about 40 or 60 feet. A perfectly good orchard tree. But it's not what we're looking for. The American chestnut tree was a canopy tree. It was a timber tree. It was a tree that grew, as you know, now to great height, 80, 100 feet or more. And we're looking to bring back a tree that has those qualities. And so simply using the Chinese or Japanese chestnut won't fit the bill. So that brings us, let's just sort of see if I can bring that down a little bit. Oh, no, not the wrong way. Let's go back. At the top, I don't know if it's cut off a little bit for you. It is for me. It says 3-burr strategy. And that the 3-burr strategy is the strategy of the American Chestnut Foundation, along with the Master Island chapter, of course. And the three Bs stand for biological control, back-cross breeding, and biotechnology united for restoration. So there you get the 3-burr acronym. And the foundation's been around for about 40 years. They didn't start out with this 3-burr approach. It was a more basic approach. And the back-cross breeding is probably the one that I would say has, from the beginning, has been a core focus. The biotechnology is now a major focus as well. But I'm going to spend some time on the back-cross breeding in a minute, because it's such a core part of what the chapters do. The biological control, by the way, just means giving the blight, giving this fungus, making it sick so that it isn't so virulent. That's basically what biological control is. And we'll touch on that. And I'm going to start, though, with the back-cross breeding. And I'm going to there's a lot of information on this PowerPoint slide. And I'm going to take a little extra time with this slide, because I want you to understand as clearly as possible in a summary way how the back-cross breeding has worked and where we currently are in the process, because the science has advanced as we've gone along. We have an understanding now that we didn't have in the beginning. But the back-cross breeding starts by taking a pure Chinese chestnut and taking its pollen and pollinating the flowers of a pure American chestnut. And when you do that, the nuts that are produced are genetically 50% American and 50% Chinese. And so you plant those in the ground and you wait 6, 7 years till they're reproductively mature. And then now you start the back cross process, because this first cross was an outcross between these two. Now you do the back crossing by taking American pollen, and you pollinate the flowers of this F1, the hybrid. And what you get are nuts that we call the first back cross. And you plant those in the ground and you go through the process again, waiting for them to be reproductively mature. And then you do another back cross, taking American pollen and manually pollinating these trees. And the process goes on. At each of these generational levels, though, there's the question, well, how do we breed in the Chinese chestnut resistance? And what happens is that at each generational level, before you do the next cross to get to the next generation, you have to inoculate every single tree with the blight. We sicken every single tree intentionally by giving it the blight. Usually you think of an inoculation, we get vaccinated. That's the kettle of fish. Here we're inoculating the trees with the blight to see how they respond, watch them over a period of time, 9 months to a year. The ones that show lower resistance, we cut them down, and only the ones that show the decent resistance at that level, do we keep and use as parents of the next generation. So that's the process, obviously time consuming. And the Mass Rhode Island chapter is down here at this level. The first orchard that I put in was one of the 3rd generation backcross orchards that the Mass Rhode Island chapter was planting at the time. And now we also have what we call seed orchards, the 4th generation backcross, the orchards. So these trees are 15, technically, they're 15/16 American, but when we test them, we find that there's variability. In some cases, the trees show a bit more the Chinese genetic material, a little bit less American. But in general, they're in that range of 15/16. Now, when all this was started, the science, the genetic science indicated that there were just a handful, less than a dozen sites on the Chinese chestnut genome that controlled for blight resistance. But as time has gone by, the science has evolved and we've discovered, well, no, that's not correct. There are more sites on the genome that control for blight resistance. And so what that means is, originally we thought by the time you get to this B3, F3 level down here, we thought, well, maybe we'd have a tree that we could start to take out of the research orchards and put out in the forest. But the fact is we need to go further, and we need to adapt the techniques. And I'm going to talk a little bit about that down the road. But this is an example of the way in which science evolves. You start with a hypothesis, you work with that hypothesis, and then additional questions come up, more information comes to light, and you adapt. So that, in a nutshell, that's the backcross component of the 3-burr program.
John, we
Yeah.
have a question. Can you explain intercross versus backcross?
Yes, let's go back to that. So up here in the early generations, we were using an American to pollinate the trees in that particular generation of the generational orchard. But here, you notice it shifts. And instead of it being an pure American tree, the pollen that we're using to pollinate this B3 tree. We're using other trees in that same orchard to pollinate each other. They were no longer doing back crossing because we've reached a level of 15/16 American, the genetic material. We don't need to go further than that. So we're letting these trees intercross. And so we do that at this third generational level, creating the first intercross nuts at this level here where you see my cursor. So that's the difference between the two. So here you're looking at a backcross orchard. The one on the left has not been selected out. The trees that we are not going to keep for breeding purposes, they have not been cut down. And the trees on the right are a section of this orchard where that roguing process has been completed. And the trees that you see on the right have been selected to be parents to the next generation. I don't honestly know what the generational level this orchard is, but they gives you a sense of what an orchard looks like at two different stages. So here we have this is Sarah Fitzsimmons, who's actually the conservation officer for the foundation now. And you're seeing the trees on the left, they don't have been selected, but you can see they have really good timber style structure. These are trees obviously with a pretty high level of American chestnut genetic material in them. And so they have good timber form and elevated blight resistance. These are actually trees at the Penn State Arboretum, and it's an orchard that's a 5th generation orchard. So it's a generation beyond the most advanced orchards in the Mass Rhode Island chapter. And the trees are selected, of course, for blight resistance, but the heredity of blight resistance, the foundation still doesn't know. And blight resistant American chestnuts now exist, but they can't be guaranteed until further testing is done on the progeny. And that may take another, 5, 8, 10 years to complete. So let's move to biocontrol. I'm not going to spend a lot of time on this because it's not a major component of the 3-burr program. It's the least significant of them, but it's still the worth sharing and a line of research worth investigating. As I said earlier, basically hypovirulence means the chestnut blight fungus becomes infected and sick. It becomes infected with a virus. And the tree on the left is a fully healthy American chestnut. It's uninfected. It's not that it's resistant, it's just not infected with the blight. And the tree on the right is showing signs of a non-lethal canker. A large canker, but a non-lethal canker where the fungus is infecting the tree, but it's so sickened by the virus that it can't fully kill the tree. And our not very, the scientific term for this is cruddy bark disease. And indeed, compared to the bark on the left, it certainly is a cruddy looking bark. The biocontrol, the hypovirulence, the foundation is pursuing it. They're doing research on it, but the chapters, including the Mass Rhode Island chapter, we aren't actively involved that I'm aware of in the pursuing hypovirulence. And it's part because the virus, it's useful for maintaining, extending the life of an individual tree, but because of the nature of the virus, it isn't something so far that they've found can be used on a large landscape the scale. So that brings us to biotechnology, the third B and the 3 BER program, and strategy of the foundation. And perhaps it's not the only important, but perhaps the most important part of biotechnology to talk about is transgenics. And transgenics is based on transgenesis, which is basically you're taking a gene, or maybe more than one gene, from one organism, and you're moving it into the genome of a target organism. And your intent is that that target organism will have a new property that can be transmitted to its progeny, transmitted to its offspring. That's the basis of transgenesis and transgenics. So there's I guess what I'd say is as part of biotechnology, in the use of biotechnology, the foundation is not just doing transgenics. One of the other things it's doing is pursuing cisgenic pathways. And cisgenics is looking at the possibility of editing genes within the chestnut genome. So for example, you could take the genes from the Chinese chestnut tree, genes that control for blight resistance, once they can be clearly identified, and then moving those genes into the American genome, American chestnut genome. That's another pathway. They're exploring that. They're working on that. But what they have, where they've made the most progress so far, where they focus most of their energy, is in transgenics. So let me share this with you. And I'm going to tell you as you look at this, to take this picture with a grain of salt. And I'll tell you why. So I think I said to you earlier that there are about 16 state chapters in the foundation, Massard Island being one of them. And they run all up and down the East Coast from Georgia to Maine. And almost all of those chapters are doing back-cross breeding, except for one. And that's the New York State chapter that for the last 15, 20 years has focused its attention on transgenics in collaboration with the College of Environmental Science and Forestry at SUNY. And they've created a blight-tolerant transgenic tree. And you can see the early laboratory-based results here. All of these trees were raised in the same environment at the same time. There are wild-type Americans on the left, Chinese chestnut on the right, and the transgenic tree, which is called Darling 54. There's also Darling 58, a kind of sibling of it. And at a certain point, all of these trees were inoculated with the blight fungus, and then they were observed. And I don't know how much time had passed when this picture is taken, but you can see the wild American chestnuts, which have the blight resistance, they are all in the process of dying. The Chinese chestnuts are doing better than the Americans, but the Darling 54 look like they're thriving, even though they've been inoculated with the blight. So on the surface, you would say, and when I first saw this picture, I had this reaction. I thought, oh my God, they found the Holy Grail. Good grief, this is it. Well, as I say, take this picture with a grain of salt. As we go through, you'll see why this is certainly a remarkable result, but the scientific process is slow and has many the twists and turns. And that's part of the story I'm sharing with you tonight. So how did they manage to produce a tree that does this in the face of being assaulted by the blight? Well, it turns out the wheat plant has a single gene called oxalate oxidase that makes the wheat plant immune to the fungus that causes chestnut blight. The wheat plant is not resistant, it's immune, which of course is even better. So they, over more than a decade, maybe a couple of decades, the New York chapter and SUNY's ECF department, they found a way to extract this oxalate oxidase gene, which is called the OXO gene, and insert it into the American chestnut genome. And it turns out that this gene detoxifies an acid that the fungus produces. And when the fungus infects a tree, the acid kills the tissues of the cambium layer, and then the fungus feeds off the dead tissue. That's the process. And the OXO gene neutralizes and detoxifies that oxalic acid. So that's the main reason why you see this result. It's a pretty dramatic result, obviously, visually. But let's keep going. A little bit more about the OXO gene. You know, when you do transgenic work, of course, you're creating a genetically modified organism. The American chestnut transgenic tree is a genetically modified organism. So it's legitimate to say, well, you know, what does that do to, you know, to have the gene from the wheat plant in chestnuts? Is that going to affect not just the tree, but, you know, animals that eat it, or, you know, we eat chestnuts? Is it going to affect us? Well, it turns out the OXO gene is widespread in our food supply. It's in dark chocolate, raspberries, rhubarb, potatoes, spinach, tofu, beans, black tea. It's just in all kinds of foods that are high oxalate foods. It's just not in the chestnut. So my hope is that knowing that, they give some research, give some reassurance to people who, you know, for understandable reasons, they raise questions about the GMO foods they entering our food supply. So when you have any transgenic organism, including the transgenic chestnut tree, there is a permitting and testing process that's very vigorous. And here you see the ESF, at SUNY, one of the orchards where it's a testing site, it's a permitted testing site under the federal government, and these chestnuts, they have to be very carefully controlled. You can't share these chestnuts with the public. You can't share them with even with the Mass Rhode Island or other chestnut chapters. They're under rigorous control until the EPA, the USDA, and the FDA go through their rigorous multi-year the regulatory assessment. And that's been going on for quite a while with that particular line, the Darling 54 tree. That tree is in what I sometimes call regulatory purgatory. And there's reason, obviously, to be very careful for the foundation to do its own testing, for SUNY to do its own testing, and then submit to the testing the process that all three of those regulatory agencies require. So that's been ongoing. We don't know what the result of that will be. But in the meantime, there's new biotech findings that have just come to light at the end of last year, and that the foundation scientists have been sharing with those of us in the Mass Rhode Island chapter and all the other chapters. And this is the information that I wanted to share with you as well. What we're understanding now is that the trees that are under the transgenic tree, the Darling line of trees, the Darling 54 and Darling 58, in the field tests they've been doing, it's come to light that there are some flaws in those transgenic tree lines. And the flaws, they are primarily that the growth pattern of these trees is not unfolding the way they expected. And they've discovered that there was early on a mistake made in that when the OXO gene was taken from the wheat plant and inserted into the genome of the American tree, it was supposed to go onto chromosome 4, if I remember right, and it was mistakenly put on chromosome 7. And that ended up silencing a gene in the American chestnut genome. And that was one problem. There's another issue that came up, and give me a moment here because it's escaping me. Oh, I know what it was. So whenever you do this transgenic process, you have to have what scientists call a promoter. And there are all kinds of promoters that scientists use when they do these transformations, these transgenic transformations. And they used a promoter that turned on the OXO gene all the time. It was a very aggressive promoter. And it turns out, the way the scientists describe it, said it was like giving the tree a fever, constantly giving the tree a fever. And the fever, so to speak, was causing a number of the problems that they were seeing in these trees in the field tests. So what the foundation has decided to do, once this has came to light, they decided to withdraw their involvement in the permitting process with the 3 federal agencies. They're no longer involved in that. They do not want, they've made a decision they will not use the Darling 54 and 58 tree in the ongoing process to restore the American chestnut. That's a major decision. And there are other, which I will tell you about in a minute, other pathways forward that are always underway. But that was the one that was most down the line, most along the path of going through the permitting process with the federal government. So that's a setback. But as I alluded to with the backcross breeding, you start with a certain hypothesis, and as you move along through the scientific process, it's inevitable that as you learn more, you discover that, oh, there's more to learn, or that this pathway isn't the right pathway. We need to back up and try a different pathway. And so that's where they're at this point with the scientific work on the Darling 54. But luckily, they didn't put all their eggs, transgenic eggs, into that Darling basket. That there were other efforts going on, and there's one called Darwin, in which they used a different promoter that doesn't turn the OXO gene on all the time, just turns it on when the tree is wounded or when the tree is under assault from something like the blight fungus. So they've done that. Also, that Darwin line of transgenic trees is one where they inserted the OXO gene on the chromosome they intended to put it on. So there are these, just within the transgenic work of biotechnology, and there are other areas of biotechnology they're working with, just within the transgenics, they have a number of pathways, including the cisgenic work, that they can proceed with. Even though there's a setback with the Darling line. So I wanted to tonight to give you the very recent update on where the science stands. This is the first talk I've given where I was able to share the updated information with anyone else. Let's see what else I might touch on here. Let's go to the next slide. So this starts to bring us back towards the backcross breeding and the ways in which the biotechnology arm and the backcross breeding arms, they can potentially begin to integrate. One of the things to take note of is that a transgenic chestnut tree, is a clone. It doesn't have any genetic diversity. And so in and of itself, you couldn't take the transgenic tree and put it out in the woods and in the forest and expect it to thrive and survive over time. It needs genetic diversity. Well, where can we get genetic diversity? Well, one of the great strengths of the backcross breeding program in the 15 state chapters up and down the East Coast is all the genetic diversity that all those chapters have bred into all of their backcross orchards, because they've collected pollen from that entire range from Georgia up to Maine. So there's the genetic diversity. So one of the questions is, well, how can we potentially, if there's going to be down the road a transgenic tree to work with, how can we make sure that the genetic diversity gets woven into the genetic material of the transgenic tree? Well, this is one of the reasons why we keep collecting pure wild American chestnuts, and why we want folks like you who they love plants and love to grow things and take hikes in the woods to keep your eye out for pure wild Americans that are actually reproductively mature. We are collecting those nuts, and one of the things we're doing with them, this is with the pure wild Americans, we are putting them into what we call germplasm conservation orchards. The Massard Island chapter has 2 germplasm conservation orchards at this point, I think. We will plant more. The Connecticut chapter has more than that and planning to plant more. And the purpose is to preserve the pure wild American germplasm. And one of the purposes for that is if down the road there is a transgenic tree, we can begin to do controlled pollinations over a period of generations, using the germplasm from the conservation orchards to begin to breed in genetic diversity to the transgenic tree. But that's also true for the backcross trees in our orchards. And there's another positive tale to tell you about the adaptations we're making with the backcross. I'll tell you about that in a minute. But the backcross trees, over a period of 4 or 5 generations, have gradually built up the more blight resistance than a pure wild American. Not a whole lot, not as much as we hoped for, but it's more. And so those trees also, because they have wonderful genetic diversity, the backcross, the orchards can be used in time, if there is a transgenic tree, to breed in the genetic diversity to that tree. So those are possibilities. Before we go to that, though, there's one more thing I want to share with you about the backcross program. One of the ways we've adapted this scientific strategy of working with the backcross trees is we've started to take the very best trees, because all these trees in every single orchard have been rated. We know where the very best among the various lines, we have where the very best tree is in a particular orchard, and the very best trees in the Mass Rhode Island orchard. There's just a handful of them in each of the chapters. You can take those trees, what we've been doing, taking those trees and starting to do best by best crosses, controlled pollinations, the best tree in one particular orchard or line, another tree in another orchard, another line. They just show that are showing high levels of resistance to having been inoculated, crossing them, taking those nuts and planting them in seed orchards where we still have space. The Granville orchard is one of the first that had the opportunity to take some of the nuts from the best by best crosses. In that orchard, there are 2 plots. That means 300 trees that are coming along. This will be their, I think, 3rd year of growth. So far, the survival rate is very high. The growth pattern is very good. And the strategy that the lead scientist at the foundation is thinking about is over a period of several generations, doing very careful crosses at each generation of best by best, and raising the level of the blight resistance of those trees well above the level that we've been getting so far from the back cross breeding program. So this is one of the ways. This is another way in which the science is evolving, and the strategies are being adapted at both in the transgenic arm and in the back cross breeding arm to the science as we learn more. So I think just about my last slide is just thinking for a moment about having listened to this talk for the last hour. If it sparks your interest in wondering how you can, make a difference here, how you could be involved in one way or another, you certainly can become a member of the American Chestnut Foundation. You'd go to their website and they sign on and let them know if you're a Massachusetts resident. You could sign up to be a Mass Rhode Island, the chapter member. You can do outreach. Renee, they approached me to do this presentation. Some of you may be part of some other group that might be interested in hearing about the story of the restoration of the American chestnut. There are several of us, quite a few of us on the board of directors of the Mass Rhode Island chapter that give talks like this. And so depending upon where you live, one or other of us they could come to your group either in person or via Zoom. And the person to contact about that, if you were interested in having a talk, is to contact Kathy Desjardins, our president, and her contact information will be on the next slide. You can plant a tree. You from us or down at Meadowview, Virginia, the national research farm for the foundation, either from them or from us, it's possible to get some trees. And from us, from the Mass Rhode Island chapter, you can get some pure Americans. Each year we seem to have some, thanks to Mark Meal and his partner, who are the 2 folks who are sort of our lead people in the chapter collecting pure American nuts. Sometimes there are backcross trees that are available, and you can get you have to pay a fee, but from Meadowview, Virginia, and the American Chestnut Foundation, the website, you can order advanced backcross trees. I think each year there are still some available. And of course, you can volunteer. My orchard and other orchards have work parties each growing season. Believe me, with 2,500 trees that need to be fertilized, the little ones need to be weeded. We have ongoing roguing, taking out the trees that we've decided we don't want to use as parents of the next generation. It's certainly not something that 1 or 2 the orchard managers are going to handle. So we're very grateful for anyone who volunteers to sign up for a work party. And again, you could contact me, you could contact Kathy, And we would find an orchard in your area and let that manager know that you might be interested in a work party this coming season. And you can also find trees. We need to find new, pure, wild Americans. And it's walking in the woods, it's fun to look for them. I drive my wife a little crazy because driving around on the roads, I look for chestnuts on the side of the road. And I've been around chestnut trees enough that just driving by them when when they've leafed out, it's fairly easy to identify them. But she prefers to say, John, you drive, I'll look. So I guess I make her a little nervous. So
John, we have 2 questions.
Sure, go ahead.
And actually also a reminder about the chestnut beet. But the first question is, what best level of blight resistance are you seeing in your orchards?
Oh, that's a good question. Well, let's take it on a scale. If you had a scale from 0 to 100, 0 means a pure wild American, the level of resistance is 0. And if you look for large surviving, what are called LSAs, large surviving Americans that are pure Americans, there are a handful of them. The foundation is studying them. On that scale from 0 to 100, those seem to be at about a 15. And by the way, 100 represents the level of resistance that a Chinese chestnut would have. So 100 at one end, 0 at the other. Large surviving Americans about 15. The backcross trees, I think at the seed orchard level, the 4th generation level, my guess is they're somewhere around 30 to 35 from what Jared, our scientist, most recently said to us. By doing those best by best crosses, and then over another generation or 2, carefully selecting at each generation the best and doing best by best crosses, his hypothesis is that we would get to around 55 to 65 on that scale of 0 to 100. And remember, the Chinese chestnut is not immune to the blight. The Chinese chestnut is resistant to the blight. It will get the blight, but at a much lower level that doesn't kill the tree usually, unless the tree is taxed and stressed for other environmental reasons. So we're not trying to get to immunity, and that scale of 0 to 100, 100 doesn't represent immunity. It just represents the equivalent of Chinese chestnut resistance.
And once it's had the blight, is it then immune or no? Like it could survive the blight and then get another infestation years later?
No, it could. I mean, you could get a Chinese chestnut or an American can get blight on a limb, but down the road it might get the blight on another limb or on the trunk. But the Chinese chestnut is capable, in most cases, of resisting the blight to a level where it can survive rather than succumb.
Gotcha. Our next question is, if I plant an American chestnut, it will eventually get the blight. Or I'm sorry, yeah, it will eventually get the blight. Where is the blight currently hiding?
Yeah, that's a good question. The blight, there are a lot of hosts to the blight. The blight, by the way, is airborne. It does not live in the soil. It doesn't kill the roots of an American chestnut. Even if it kills the tree above ground, the roots continue to thrive and put up root sprouts. And that's one of the reasons over the years we have so many these surviving small chestnuts up and down the native range, is because chestnuts happen to be vigorous root sprouters. But it's because it's airborne, it's kind of everywhere, but there are certain hosts. Oak trees are hosts to it, I believe. I believe sumac is. There are a variety of plants and trees that are host to it, but don't succumb to it, aren't vulnerable to it.
So we're almost out of time. We have one more question, but we also, I think a lot of people are wanting to hear about the chestnut bee. So maybe let's talk
Okay.
about the chestnut bee, and then that last question, if we have a few seconds, we can ask that.
Okay. So I ran across an article not long ago. It was in the newsletter of the Connecticut chapter of the American Chestnut Foundation for the spring issue of 2022. And they were talking about a chestnut bee, which is its taxonomic name, I think, is Andrena rainey. And it was thought to be extinct due to the fact that, the chestnut is functionally extinct. But there's a fellow by the name of Michael Veet who has specialized in doing research on the chestnut bee. And he teamed up with the Connecticut chapter and the Mass Rhode Island chapter, and they set up a survey, and they wanted to assess the distribution and abundance of this chestnut bee on chestnuts in orchards and also on other sites. And the survey, they thought, would also help determine the diversity of bees and other insects visiting chestnut tree flowers. They ended up collecting from 55 visits on 27 sites around southern New England, including 11 in Connecticut. They collected a total of 685 bees from 61 species. That astounded me when I read that. And to the delight of everyone involved in the survey, they found the famous chestnut bee once again, and they collected from 2 sites where they had previously seen the bee. And then, let me see this here, make sure I say it right. There were 38 chestnut bees found at the Lockwood Farm in Connecticut. 3 were found at Mount Ella in Massachusetts. 3 more were found in a new location on Mount Tom in Massachusetts. And that all these bees were collected on multiple specimens of chestnuts and chinkapins, both domestic and imported. And their preliminary survey, they thought, would help scientists better understand this shy bee and develop protection plans to see the population thrive. So I just thought that was a great news story and survey, and I couldn't think of a better group to share it with than you all. So I thought I'd wrap up the talk with that.
Thank you. Thank you so much, John. This was really great. So much wonderful information and quite a saga. There is one more question. If you have, it sounds like it's a yes or no, but the question was, are you using synthetic fertilizers on your trees?
The answer to that is yes. In the beginning, we will use a 10-10-10 fertilizer with the young trees. We also do the irrigation for the trees when they're young. Once they get to a certain size, we aren't using the synthetic fertilizer.
Thank you so much, everybody, for coming tonight. You will get a recording of this in the next day or so. I apologize, my computer had a catastrophic failure in the middle of this presentation. Thank goodness John was a co-host, so it didn't end the meeting. But I don't actually have the links for our brown bag lunch or our next meeting, and I apologize, but I will email those out again. So thank you, John, so much. You have a question. Yes, go ahead.
Yeah, I noticed that Michael Veet was actually on the call. I saw his name a moment ago. I don't know if he's still here. I just wondered if Michael wanted to add anything to what I said about the chestnut bee, because he's the researcher who specialized in it. Michael, do you want to say something? I don't know if he's still here. He may have left. Okay. I couldn't resist asking him.
Thank you, John. This was great. And thank you, everybody. And we will see you next month.
Okay. Thank you all.
Bye.